I'm Ryleigh Newman,
And I'm Amber Wolf.
and this is the research into heritability of ADHD.
So, a brief overview. What is ADHD?
ADHD stands for Attention Deficit Hyperactivity Disorder, and it is a neurodevelopmental disorder commonly seen in school children.
This is characterized by difficulty in problem solving, paying attention, and hyperactivity. It's also attributed to a variety of causes and is not 100% understood yet.
It is classified into three types: inattentive, hyperactive/impulsive, and combined. It is believed to be linked to genes that are known to cause other types of mental issues.
In this presentation, we will discuss the current research findings of whether ADHD is an inheritable condition and whether current research into the heritability of ADHD provides substantial evidence to argue this, despite still not being absolutely proven.
For example, in a set of highly constrained genes, roughly 3,000, that show high intolerance to a loss of function, many showed significant association with ADHD, according to one of our research papers of reference.
Twin and methods of studies. Studies involving twins, both monozygotic and dizygotic, also known as identical and fraternal twins, are often compared with non-twins or only children to examine heredity. These are usually done by looking at those who were adopted.
International studies have found ADHD heredity rates ranging from 70 to 90%. The likelihood of ADHD in first-degree relatives, such as full siblings and parents, is notably higher than more distant relatives.
Twin and adoption studies suggest that the condition is more likely to be inherited by children with a genetic connection, with the heritability estimate ranging from 60 to 90%, with most studies typically finding around 70 to 80% heritability.
Comparison between identical and non-identical twins helps determine the degree to which genetics might contribute to the observed trait.
ADHD family and twin studies show genetic connections shared with other conditions like antisocial personality disorders and behaviors, cognitive impairments, autism spectrum disorder, schizophrenia, bipolar disorder, and major depressive disorder.
Purpose of understanding linked conditions. ADHD overlaps with 43 different phenotypes, including major depressive disorder, anorexia nervosa, educational outcomes, obesity-related traits, smoking habits, reproductive success, insomnia, and mortality. Genotypic background, developmental problems, and depression. The loci on chromosomes 7 and 10 each have credible regions of association localized to a single gene with limited additional annotations.
In the chromosome 7 locus, FOXP2 encodes a fork-head winged helix transcription factor and synapse formation, as well as neural mechanisms mediating the development of speech and learning.
On chromosome 10, the ADHD association is intronic, located in SORCS3, which encodes a brain-expressed transmembrane receptor important for neuronal development and plasticity. It has previously been associated with depression.
For the phenotypic background, on chromosome 7, the gene FOXP2 is involved in specific developmental disorders related to language and learning. This is commonly associated with ADHD with a 7 to 11% prevalence, which impacts crucial mechanisms for formation. Poor language skills are also frequently linked to this.
Chromosome 10 expresses genes related to transmembrane receptors crucial for neuronal development and plasticity. These genes have previously been associated with depression. Genotypic background. Chromosomes 12 and 15 exhibit more biological annotations that provide support for their significance in certain traits or conditions.
On chromosome 12, the locus encompasses DUSP6 and contains an annotated missense variant in the first exon and an insertion near the transcription site, although neither of these is actually the primary or lead variant.
Chromosome 15's locus resides in SEMA6D, and the majority of variants within the credible set are linked to the expression of this gene in fibroblasts.
The phenotypic background. Chromosome 12 encodes the dual specificity phosphatase that potentially regulates neurotransmitter homeostasis, impacting the dopamine levels in synapses. This regulation is relevant as commonly used medications for ADHD target the dopaminergic system, which increases synaptic dopamine availability.
Chromosome 15 harbors SEMA6D, active in the brain during embryonic development.
This is believed to contribute to neuronal wiring, and variants in this region have previously been linked to educational attainment.
More diverse regions of interest. On chromosome 1, the primary variant lies within an intronic region of ST3GAL3.
Missense mutations in this area have been proven to cause autosomal recessive intellectual disability. Additionally, an intergenic variant within this locus has been previously linked to schizophrenia.
Chromosome 5 is associated with LINC00461, a function that remains unclear but shows highly localized RNA expression in the brain. Variants of this genomic region have been connected to educational attainment.
A significant SNP across the genome is situated in MEF2C-AS1, which is particularly intriguing due to past associations between MEF2C and severe intellectual disabilities. These include cerebral malformations, depression, schizophrenia, and Alzheimer's disease. However, this association is not supported by a credible set analysis.
Competitive gene-based tests were conducted on specific target genes. However, 3 separate sets of genes downstream from FOXP2 didn't show any significant association with the condition.
The absence of association could be due to several factors, such as the unknown functions of FOXP2 that contribute to the risk of ADHD, insufficient statistical power to detect relevant downstream genes, or possibly that only a small subset of biological functions that are regulated by FOXP2 are relevant to the development of ADHD.
Purpose of understanding. Replication studies meta-analysis. In the replication studies, out of the 12 loci examined, 4 were consistently significant across all 3 studies. These include ST3GAL3/PTPRF, FOXP2, SORCS3, and an intergenic locus. In the replication meta-analysis, 3 loci were consistently significant in 2 of them. Notably, CADPS2 was the main locus and has been recently identified in autism spectrum disorder as a novel locus that is shared with educational attainment.
Areas of improvement to consider. Genetic correlations might not universally apply to all settings, indicating worse external validity as it is less generalizable. Alongside this, in the replication studies, some results showed stronger associations while others had weaker correlations. These differences could stem from the method of phenotyping, whether the data is self-reported or sourced from medical records, and the diversity within the study population, considering factors such as socioeconomic status and potential underrepresentation of individuals with poorer educational outcomes.
In one setting, the correlation might appear stronger due to precise medical record data and a diverse participant pool. Meanwhile, in another setting reliant on self-reported information and with a less diverse, potentially underrepresented population, the correlation might seem weaker. These disparities underscore the impact of varied phenotyping methods and the diversity of the study population on the observed genetic associations.
Genetics and the environment. Behavioral genetics often investigates the interplay between genetic and environmental factors in influencing conditions like ADHD. In gene environment studies, interaction between environmental and genetic factors is a crucial aspect of ADHD pathogenesis. While prenatal environmental risk factors might have a smaller impact individually, their combined effect with genetic predisposition can contribute to the condition. Studies have supported various environmental factors influencing inattentive or hyperactive impulsive symptoms, with psychosocial factors notably influencing inattentiveness and biological factors having a greater impact on hyperactive impulsive symptoms.
However, not all studies align on the concept of gene-environment interactions. For instance, one study showed a strong link between early life exposure to environmental risks and specific neurodevelopmental risk alleles, while another study found no significant interaction between a genetic allele and maternal smoking as an ADHD risk factor. This variance among studies emphasizes the complexity of understanding how genes and the environment jointly contribute to ADHD.
Theoreticals and possible future research. Research into the genetics behind ADHD and potentially finding enough evidence to confirm matching genes allow for the possibility of looking ahead into what future treatments can look like.
For example, in cases where ADHD may be incredibly severe, in the future, perhaps, methods of gene therapy can be used to help improve the symptoms.
As of the current dates of writing, November 11, 2023, scientists believe that CRISPR technology can treat ADHD. However, no research has tried this yet.
Summary of the genetics behind ADHD. So, we know that ADHD is heritable and primarily caused due to genetics, as it is more commonly seen in first-degree relatives of those who have it. While there's evidence of genes that are thought to be linked to ADHD, it's not certain what their full extent is, as they may lead to more susceptibility rather than the condition itself.
When comparing 3 research articles from the past recent time, we can observe genes such as FOXP2, SORCS3, and DUSP6 located in or near several of the loci suggest involvement in the neurodevelopmental processes relevant to ADHD.
While the roughly 12 identified loci present compelling findings, they represent only a small fraction of the common variant risk for ADHD. These effect sizes fall within the observed range for common genetic variants in other highly polygenic psychiatric disorders, such as schizophrenia.
And here's our works cited. Thank you very much.
Thank you. Booyah!